What is ecological stoichiometry? That's the question I want to answer today on Science Spotlight 9, Ecological Stoichiometry. Dr. Mark Williamson, let's get into it. So, to start, let's introduce the topic and define our topic. So we can pull up from Merriam-Webster the adjective ecological relating to the environment of living things. That's useful. Okay, relating to the environment of living things. And then stoichiometry comes from a lot of times chemistry, quantitative relationship between constituents of a chemical substance. You think of elements. So something to do with quantitative relationships between elements in an ecological or environmental living things concept. And indeed, when you find a definition in a book we'll talk about in a moment here, of ecological stoichiometry. It's the balance of multiple chemical substances in ecological interactions and processes, or the study of this balance, also sometimes referred to as the balance of energy and materials. And particularly the chemical substances will be elements. The foundational text for ecological stoichiometry is the 2002 book, Ecological Stoichiometry, The Biology of Elements from Molecules to the Biosphere by Robert Sterner and James Elser. So this is sort of the foundational text laying the groundwork for ecological stoichiometry in the last 25 years. Some of the concepts are not didn't originate there, but sort of this is the sort of the synthesis, the modern building of it and launch from there. So the book, covers, as I said, a wide range of things, starting out with sort of the basics, stoichiometry and homeostasis, biological chemistry, going on to stoichiometry of autotrophs, animals, look at imbalanced resources and growth, stoichiometry of consumer-driven nutrient recycling, stoichiometry in higher levels like communities, and even big scale like ecosystems in space and time, with a final sort of recapitulation and integration. Now, there's too much to go into every detail of the book, but certainly if you want to learn about ecological stoichiometry, you couldn't do worse than just simply read this book. I read it several years ago in graduate school. So what I think I'll do here is just sort of go over some of the basics of this book, the first two chapters, and then the chapter 9 recapitulation. So in the first chapter, Sean goes over the idea of stoichiometry, homeostasis. A lot of it is looking at not just the elements in, say, living things, but the element balance. So this is a great figure, is the abundance of elements in humans relative to carbon, carbon being one of the most abundant elements, carbon-based life it is. And so the y-axis is actually log carbon to that element ratio. And so the bigger they are, that means that actually the less the lower their abundance is, there's more carbon to that element the higher you go up. So it turns out there is a little bit more oxygen than carbon, but then as you kind of go up, these can get rare and rare. So like things like copper and iron are not iron, copper and things like copper, selenium, other chromium, a lot of these metals are trace elements. And a lot of ecological stoichiometry, deals with carbon to other element ratios, especially things like nitrogen and phosphorus being very important. And we'll see those come up again and again. Another great figure in this chapter is 1.4, looking at, they're really organisms need to take in elements, need to take in nutrients and build their bodies. And so there's going to be this balance between the research stoichiometry, so what they take in to eat, and the actual consumer, what their elements are made of, what ratios of carbon, nitrogen, phosphorus are made of. Now, if there is no difference, you get sort of this line here: you are what you eat, no homeostasis. But if there's some sort of imbalance, one way or the other, the organism has to balance for that. They have to engage in homeostatic regulation to get the right elements. And I think that's a really important concept of ecological stoichiometry. Organisms need certain elements, and there'll be different values, different ratios in their environment, and they need to find ways to get it, some and not others. And that has effects on growth rate, species dynamics, what species can live where, how different niches work. So a lot that goes into this, surprisingly. Another great figure is figure 11. Brings up a very famous thing in ecological stoichiometry that predates, to 2002, which is the Redfield ratio, which is actually the constant or consistent atomic ratio of carbon, nitrogen, and phosphorus found in marine phytoplankton and throughout deep oceans. It's essentially 106 carbon to 16 nitrogen to 1 phosphorus. So kind of shown here. It's shown to be fairly consistent across various measurements, though not perfect. And so that's an important concept in ecological stoichiometry. And I think this is back in Redfield ratio is like 1969, so it quite a bit predates the actual Stern and Elzer book. Another useful figure in this chapter is these different sort of setup of ecological stoichiometry with theorems and axioms, and how they interrelate with each other, and how you can answer a lot of really interesting questions with ecological stoichiometry. Starting with, say, the theorem that elements limit growth. Well, with that theorem, there's going to be things that go into, axioms that build out of that. So, there's an organizational pattern in stoichiometry, there's common stoichiometry and protoplasmic life, etc. You can see that with sort of this building up of this conceptual framework, you start to answer and test really interesting questions. All right, next chapter of Stern and Elzer's 2002 book, Biological Chemistry, Building Cells from Elements. This really goes into the fact that, oh yeah, biological life has different components and different elements go into that, and that really has an effect. For example, table 2.1 is like functions and chemical elements. So there's different elements like hydrogen, oxygen, carbon, and others, include nitrogen and phosphorus, that are all structural, that you need to build tissues, skeletons, shells, etc. There's electrochemical elements like sodium, potassium, chlorine that create free ions. There's mechanical, like muscle contraction, catalytic, different types of catalytic. And so different elements are needed for different things. And I think table 2.2 is really cool because then that goes into, well, there's different classes of biological materials, in the cell, and they need different things. So like protein, the thing in parentheses means that only certain forms. So in protein, most all protein needs carbon and nitrogen, though only a few need phosphorus. So protein is much less, doesn't really need very much phosphorus, while nucleic acid needs all three, carbon, nitrogen, and phosphorus. Lipids really just need carbon, not very much nitrogen and phosphorus, and so on and so forth. And so what different things in the cell need to be built, will have different elemental demands. And I think this is a great figure. Talks about the shows the percentage of nitrogen and phosphorus of different biomolecules. So as you go up on the x-axis, phosphorus demand goes up, and on the y-axis is nitrogen demand. You can see here that things like protein really don't need any phosphorus at all, and really just high in nitrogen, whereas something like phospholipids need phosphorus, but really almost no nitrogen. But a lot of things are in between. Like, for example, nucleic acids are a good intermediate where it needs both phosphorus and nitrogen in decent amounts. And then going back to chapter 9, where it sort of recapitulates and it integrates what the book talked about. Some important pieces in the recapitulation, not all of it, but I think these are helpful points, that really ecological stoichiometry covers a lot of areas. Its topics ranging from mundane to exoteric and staggeringly small, so subcellular, to numbingly large, huge, like the biosphere, and pointing out that the ratio of carbon, nitrogen, and phosphorus, that stoichiometry is the most important. Now you can look at stoichiometry of other elements, but these are the most important and traditionally been explored the most. And another point that maybe isn't obvious is that stoichiometry in metazoans, so that's animals, is much more tightly constrained than in autotrophs, so like plants, algae. And so what organism you look at will depend on what sort of stoichiometric flexibility is even present in the system. And some important integration points. As biological research has become more sophisticated across the decades, it's become more fragmented. And so finding ways to actually unite these fragmented pieces is really useful, and it's actually a thrilling challenge to reconcile those knowledge. And ecological stoichiometry is one such method. And so stoichiometry can be thought of as a simple rule to explain biological complexity. So you can take carbon to nitrogen to phosphorus ratios and answer really important questions about biological complexity with a seemingly simple sort of measurement. All right, important figure in this last chapter is looking at a sort of a stoichiometric number line of N and P ratio. So on uh, the left over here, this is sort of the nitrogen. So it goes sort of infinity for things like proteins, goes to infinity for, or zero, that is no nitrogen, all phosphorus, over in this side for things like uh, polyphosphate. And a lot of like the actual biological things are fairly constrained here. You see the Redfield ratio here, whereas some more abiotic things like Earth's crust is farther out here, or runoff from unfertilized fields. And so you can see there's definitely a range of NP ratios across both biotic and abiotic systems. And then I think a figure number two is a great composite figure, really highlighting the fact that stoichiometry really does range from genes to ecosystems. You can start at the gene level, look at the ratio of N and P, or the percentage of M and P of biochemicals, moving up to organelles. You can do things with organism growth rate, how they how consumer and research stoichiometry interlock. You can talk about consumer herbivores, autotrophs, the fate of carbon, herbivore growth, consumer herbivore nutrient recycling, even consumer herbivore autotroph dynamics. And so it really ecological stoichiometry does range a huge range of what you can look at, what kind of questions you can answer. All right, so with that, see the Ecological Stoichiometry book in 2002 by Stern and Elzer was really important, but that's certainly not the only thing. A lot of work has been done from there. For example, there's recent advances in ecological stoichiometry, insights for population and community ecology, back in 2005, with objectives to review recent progress. Um, from studies after Sterner and Elzer, identify gaps, and then look at promising directions. There, a little bit later, there was the 2013 paper, Ecological Stoichiometry, an Elemental Approach Using Basic Principles. We see Elzer and Sterner as part of this paper as well. And I really like, one of the points I really like from this paper is that ecological stoichiometry really started as a very small niche perspective, looking at Daphnia, so water fleas in aquatic systems, but it's since widened into many organism types, many ecosystem types, and the questions it address has broadened. And so this paper addresses some of the development in especially in the aquatic sciences over the past 10 years, and how ecological stoichiometry has provided novel insights into things like genomic, proteomic, and cellular responses, and as well as looking at large-scale effects related to biogeochemical couplings at the entire ecosystem level. Other work, there's a good paper in 2015, the application of ecological stoichiometry to plant microbial soil organic matter transformations. So it expanded beyond just simply aquatic systems. Looked at things like, nitrogen and phosphorus in the soil. Looked at ratios across different constituents of the soil, and different effects of research stoichiometry and environment on especially like microbial community structure. Really, because there's an inherent link between biogeochemistry and the structure and process within food webs. And so this paper's review looked at especially the structure and function of heterotrophic microbial communities and how research stoichiometry matters. There are some general patterns that it suggested that Latitudinal gradients of soil and litter stoichiometry are reflected in microbial community structure and function. That second, research stoichiometry may cause changes in microbial interactions in community dynamics. And third, that there are global changes that alter the carbon nitrogen, carbon phosphorus, and nitrogen phosphorus ratios of primary producers. And then they end by arguing that ecological stoichiometry provides a framework to analyze and predict such global change effects at various scales. And something they bring up, and for example, it's abbreviated here, an important concept that comes up again and again in ecological stoichiometry, like the Redfield ratio, is something called the growth rate hypothesis, or GRH. And it hypothesizes that elevated demands for increased allocation to phosphorus ribosomal RNA under rapid growth drives variation in the phosphorus content of many biota, especially in microbes. And so we know that, yeah, so nucleic acids are fairly phosphorus heavy, demanding, and ribosomal RNA is needed for growth. So there's going to be this interplay between how fast an organism can grow, especially a microbial organism can grow, and the amount of phosphorus that it can take up. And then here I'll kind of show some of these figures in a little bit greater levels, looking at the different effects of um, limitation of nitrogen and phosphorus, the effect of temperature, and the interplay between local and global mechanisms. This figure I really like here, looking at uh, carbon and nitrogen ratio, carbon and phosphorus ratio, nitrogen and phosphorus ratio across different organisms. You can see, wood has a much different carbon and nitrogen and carbon and phosphorus ratio than soil bacteria. And so how these all interact really matters. And then, yeah, this figure six is really nice in sort of the big scale effects across things. We have external drivers of, say, environmental conditions on the microbial community composition and how that affects their stoichiometric ratio and how that affects all these other things. And so these sort of high level graphics are really helpful to start to piece together how all these things interact and what interesting questions you could maybe pose and try to answer. All right, and so it didn't stop there. There's been continual progress and advances in ecological stoichiometry. For example, there's this editorial, Progress in Ecological Stoichiometry, and we see Sterner and Elzer pop up again here. This was in 2018. This is actually part of, the introductory part of a collection of 34 articles, labeled in Frontiers on progress in ecological stoichiometry. A great quote from this article or this editorial is, since pretty much everything that is a thing is at its core elemental, novel applications of the stoichiometric framework await. So ecological stoichiometry only has more to give, more utility as we think more about it to answer questions in biology and everything else. Covers some of the topics that will go into these articles, like microbes, primary producers, herbivores, pathogens, topics on crossing organizational scales, and even going on beyond CNP, looking at other elements and their ratios. Uh, this editorial talks about the societal relevance of ecological stoichiometry, whether it's agriculture or wastewater treatment, even ecosystem services, and some future contributions that it highlights that ecological stoichiometry could be used in human health for food nutrition, looking at understanding disease and the human microbiome, sustainable agricultural, and even elemental cycling. So there's a lot that can still be done in ecological stoichiometry. It's really a rich field for inquiry. Another editorial a little bit later, just with Elzer here, not Sterner, and of course the co-authors, all the other authors. This Advance in Ecological Stoichiometry was another collection in Frontiers called Advances in Ecological Stoichiometry. Unsurprisingly, it was just 6 articles, and it really was talking about ecological stoichiometry as being very highly disciplinary, and it covered these things called the Wood Stoic workshops. They're held every 5 years on ecological stoichiometry. And the fifth one was in 2024, held in Germany, and actually resulted in a publication of 5 novel scientific articles. Those are the other 5 articles in this 6 article collection. Jones et al. Looked at the fate of elements within phytoplankton cells. Olson et al. Looked at phytoplankton stoichiometric traits. Shinon et al. Looked at a review of phytoplankton mixotrophic metabolism. Bradley et al. Looked at dynamic energy budget model. And Reeve et al. Had a review of optical remote sensing to quantify elements in the environment. Another, backing up a couple years in 2021, a really great paper called Recent Advances in Future Research in Ecological Stoichiometry, which I think it's helpful to go over. So it was a summary of recent advances, some general points. It covered the explosion of terrestrial ecosystem studies, the useful tools that have arisen for use in ecological stoichiometry, like metabolomics and metatranscriptomics. The idea that looking at a simulated matter nitrogen to phosphorus ratio probably matters more than just what's taken in. So the ratio of food NP ratio is less important than the actual assimilated matter. Different advances have generally shown the support of that growth rate hypothesis. It's an H here. And to some extent, still the Redfield ratio, though there's not always as much agreement with the Redfield ratio. You can see places where that's violated. A great analogy this paper brought up about bricklayers and bricks. So that's like amino acids and proteins, and bricklayers, so that's phosphorus-rich ribosomal RNA. They're both needed to build the walls of a house, so that's an organism cell. So even if there's many bricks available, the walls can't be built without the brick layers. So even if you have all the elements you need to make proteins and from amino acids, you still need ribosomal RNA, which is phosphorus rich, to get those together. Another general points, the effect of low and high trophic level. So both predation and nutrient quality matter. And a really interesting correlation between element ratio and phylogenetic distance. Turns out in over 90% of recent studies, there's a significant correlation between species elemental composition and taxonomic and phylogenetic distance. Some emerging paradigms that this paper brought up. There's the multivariate productivity diversity hypothesis, MPD, which is a multifactorial equilibrium that relates the diversity of phytoplankton communities with the amount of resources, mainly phosphorus. There's the biogeochemical niche hypothesis, or the BN hypothesis, which posits that there's a relationship between anatomical and functional traits of organisms and communities and their elemental compositions. There's the study of ionomics, so the interaction between bioelemental content and electrical charge, mostly things absorbed from soils and their impact on plant health, food quality. And then there's even ecological enzyme stoichiometry, looking at the stoichiometry of enzymes in the environment. And I think an important point is this paper brings up, why ecological stoichiometry might be relevant to human health, like looking at nutrient deficiency, gut microbial community composition, human illnesses and disorders, and even the idea that there are found in cancer cells that they are more phosphorus rich than normal cells, and so they might be phosphorus limited, and so that might matter to how to treat and deal with cancer. And really they end with the idea that there's an immense potential encompassing aspects of ecological stoichiometry on human safety and socioeconomics. Some important figures from this paper. Box 1 has classical paradigms on the left here, things like growth rate hypothesis, community structure, Redfield ratio, and some new challenges that stoichiometry can try to answer. Like on are these ratios uh, more relevant in terrestrial marine ecosystems? You know, dealing with other elements ratios, drivers of global change. What about flexibility or shifts in ecosystem stoichiometry and how that affects human health and wellness? And then there's this figure here with, different achievements. So like things with a V is consistent support, but then there's question marks like where open scenarios. What's the effect of food stoichiometry in human health and wellness? Um, uh, when it comes with things with like drought, how does that affect CMP ratios, etc. And as we go forward, I found a really important paper in ecological stoichiometry that I think is helpful, that helps give us caution. It's the 2020 paper by Dr. Isles, The Misuse of Ratios in Ecological Stoichiometry. And I think anyone doing ecological stoichiometry should take this in mind. Ratios, so CNP, CN, NP, etc., they're heavily used in ecological stoichiometry. But ratios by their nature don't respond symmetrically to changes in the numerator and denominator, and they're not normally distributed, so you can't treat them like they are. So if you're calculating means, variance, or linear slopes based on ratios and you don't transform them, they're going to have biased results. And so actually, I was reviewed not over 100 studies in ecological stoichiometry and found that actually 93% had at least one error with ratios. And so the advice is you actually really need to immediately calculate log ratios, which don't have that bias from those individual observations, and keep ratio data in that form for all subsequent analyses. So whether you're doing averages, models, graphs, etc. So a good example here in figure 2 is NMP ratios across uh, time of a hypothetical data set. The untransformed is A. You can see here that it's unlog transformed, and it looks like it's only um, sort of phosphorus limited because there's a lot of nitrogen up here and not as much here. But if you actually log transform it, it's clear that actually both uh, can be limited across the season. And so that's really important, I think, to keep in mind. All right, with that, I kind of want to have a contemporary study that kind of shows like what can be done now and today in ecological stoichiometry that's interesting. So I found this paper called Microplastics Affect the Ecological Stoichiometry of Plant, Soil, and Microbes in a Greenhouse Vegetable System, 2024. So yeah, I think it's very contemporary. Microplastics are in the scientific literature a lot now. And so yeah, adding that to the soil is going to change stoichiometry. So this paper looked at that, had sort of a structural equation model, looked across different things. I think the important take-home point was some of these figures here, and in fact reinforces the caution we talked about here. You can see that there is four different treatments. The CK was a control, and then these PE 1 through 3 are various levels of microplastics that they added to these pot experiments of bok choy. And we see here that uh, the top one is the carbon to nitrogen ratio and the carbon to phosphorus ratio. You see that goes up as you add more microplastics. So you add more carbon to the system, and so the ratio of nitrogen and phosphorus go down. Though you notice here that these are not log transformed, so you should really be log transforming these as the caution showed you. But um, they looked at the soil, they looked at soil microbes, and they looked at the plant's shoot and root, at least for carbon and nitrogen. And so that's something you can do with ecological stoichiometry today. So I want to end with what I think some of the frontier is that I didn't hear mentioned. Now, I think things with human health, global sort of cycles, those are all relevant, but something that I didn't see covered yet that I think is going to be really important is space agriculture. Because if um, we're going to have like long-term space occupation on the moon or farther out, Mars, whatever, you're really going to need to deal with ecological stoichiometry in growing things like crops in space. You have to deal with the stoichiometry of like substrate, like lunar regolith, the plants itself, or even microbes that you try add to turn the regolith into living soil. And so those ratios of carbon, nitrogen, and phosphorus are all going to matter, and as well as other elements. In fact, I even tried to start doing some uh, initial investigations about like what what needs to be done with space agriculture and ecological stoichiometry, like thinking about, well, where do plants get elements? And so where will they need to get them in space? And what's like the ecological stoichiometry of growing plants in space? So obviously a lot of this is unfinished, but thinking about um, the level of nitrogen, oxygen, even carbon dioxide in atmosphere. What about the International Space Station? This is like lunar regolith. So what levels of those? Uh, what levels of plants? Where are they going to need to get it? Because that's going to be questions and problems that are going to be solved in space agriculture. And I think ecological stoichiometry is going to be really important. So that's a good example of some future research that is underutilized right now. All right, with that, here's references that should be found in the description of this video. Thanks so much for joining me. I really enjoyed going over ecological stoichiometry. It got a little long, but I think well worth our time here. With that, please like, subscribe, check out Transcend for more things, and please leave a comment on what you like the most and what topic I should cover next. With that, I will leave you here. Remember, everything that is in essence a thing is made of elements, and so you can do ecological stoichiometry with them. Signing out, Mark Williamson.